A very warm welcome to all the chairperson, speakers, delegates, and my dear colleagues. I am Anamika Kumar Yanuja, a research scholar from SGPGI Lucknow. Currently, I am working on the area of clinical immunology and myositis. I feel privileged to address you on the behalf of the conference committee and welcome you all to the 37th Annual National Conference of Indian Rheumatology Association. We have opportunity to explore our knowledge and understanding on wonderful occasions like these. We are eager to learn and what the brightest mind have to say about this incredibly timely topic of rheumatology research, and this is the main focus of the session. We will learn about the topic that are mostly unexplored in daily life: environment, climate change, rheumatic and musculoskeletal diseases. Moreover, we will emphasizing how these elements are crucial to the rheumatic and musculoskeletal diseases. We are highly obliged to have with us Dr. Zoya Zaidi and Dr. Alpana Parmar as our chairperson. Dr. Zoya Zaidi is a physician specialist in clinical rheumatology and rheumatology. She is having 35 years of experience in treating arthritis patients. She provides rheumatological and clinical immunological services to the patients not only from the India but also from the patients in Dubai and USA as well. She has been awarded with the best all-round students of the Southeast Asia region in the year 1972, 73, 74 and 75. Also won the first prize in the paper on viral hepatitis B and its association with the Australia and region in International Students Conference 1974, Moscow. Dr. Alpana Parmar is the best rheumatologist in Surat and passed her MBBS and MD from the Baroda Medical College in 2004. She then worked at Smimar Medical College as Surat Associate Professor of the Department of Medicine. In 2013, she joined PD Hinduja Hospital in Mahim, Mumbai as a fellow in the Department of Rheumatology. And I now request Dr. Zoya Zaidi and Dr. Alpana Parmar come to the dais and chair the session. We would now initiate with the keynote lecture session. We will having three distinguished speakers with us: Dr. Lars Glasgow, who will be delivering his keynote presentation on the topic, the impact of climate change and environment on rheumatic diseases. Dr. Lars Glasgow is a Swedish physician, immunologist, and rheumatologist known for the research into the genetics of autoimmune disease such as rheumatoid arthritis. In 2013, he received the Graf Ford Prize for polyarthritis research. Next, we have Dr. Vit Das here with us, and he will be enlightening the session with his talk on vitamin D, the master orchestra player in RMDs. Dr. Vit Das has earned the PN Berry Fellowship in 1995 and receives training in rheumatology at SGPGI Lucknow. He received more than 40 awards and felicitations by various organizations, including the Odisha IRACON 2011 and Odisha Citizen Award. Last but not the least, we have Dr. Rudran Chatterjee. He will be presenting on how real is the effect of UV rays and seasonal variation on RMDs. Dr. Rudran Chatterjee is Senior Resident at SGPGI Lucknow. I now request all the speakers to come to the dais. Now I would like to invite our very first speaker, Dr. Lars Glasgow, to come to the dais and deliver his talk.
So, thank you again for inviting me to this fantastic meeting. And I was given this challenging title by Dr. Danda. And while contemplating this task, I scrutinized the literature.
Which one is this?
And found out that not so much was written about climate change and musculoskeletal diseases, but much more about climate change and the impact on health. So I will divide my talk into parts. First, a bit of the more common things, which I think is very much urgent for all of us, and then some specific things concerning effects of climate change on musculoskeletal disease, in particular rheumatoid arthritis. So, this is the plea from one of the youngsters in my country, Greta Thunberg, who started a worldwide movement called Fridays for Future, where lots of young people all over the world were engaged in what were considered as their future. And of course, this is a most urgent issue for all of us. This is the most recent report, a rather large one, from the Lancet, where they've taken a big picture on what is the impact of climate change on health in more general sense. It's a big report over many different areas. And this is what they summarize, that climate change affects the health in very many areas of our life: temperature, seasonal change, extreme events, biodiversity loss, effects on food, effect on heat. And this is a very useful report, I found out, so I recommend it for everybody to consider and read. And then why, this is also from this same report. What they summarize is what we actually know, but not always consider about what is happening around us, about the disruption of water and ecosystems and the crisis of malnutrition that may happen. Examples. This is an example of the type of heat waves, which will of course affect everybody. But what they have taken particular emphasis on in this report is the impact on children and the impact of elderly. These curves show the numbers of heat waves more in general, the blue line, and more on the impact for children and for elderly people, which is even larger. This is another slide from this same report showing the increase of danger days for fire. And they show how that has increased. And notably, India happens in this report to be the area of the world where obviously the risks for fires has increased the most. This comes a bit closer to what we'll discuss specifically for the musculoskeletal and rheumatic diseases. This is the mortality related to different type of emissions, both in general causing the CO2 emission, but more specifically what is the impact of the particles from different type of emissions. And they have taken what they call the PM12 2.5 particles that they measured. And as you see here, they have what they call countries with different levels of Human Development Index. India happens to be in the medium. And in this case, they see what is the impact on mortality from different power plants, from industry, from agriculture, and so on. And what you see here is that the impact on mortality has not moved a lot from the medium income or medium development index countries, whereas it has been reducing a bit, but not enough, in the very high HDI countries, which Sweden, where I come from, happens to be. Now, this is just another illustration which I got from my friend, professor of global health in Karolinska Institutet, Stefan Svartling. And what he says is that these effects on particular matters impact a lot on inflammation and also in this case on cardiovascular disease. And these are studies from Thailand, from the United States, and the lowest one from India. They summarize what happens with inflammation in this rather complicated picture, which should emphasize what happens in the respiratory tracts, with lots of impact on quite well-known molecular studies causing different kinds of inflammation, which of course cause respiratory disease. But what we'll see in a second, in a few minutes, we will see how that may also affect the rheumatic diseases, not only directly the respiratory diseases and cardiovascular disease. Of course, I cannot go beyond the infectious diseases, which are of course affected a lot. This happens to be the slide that they have on dengue, but of course also chikungunya and Zika virus of the arboviruses follow the same pathway. And this shows the increased replication rates for the reproducibility index for, in this case, for dengue, but it's very much the same for the other arboviruses. So these are things which I think we all have to take into account when thinking about health in general. More particularly, when we think about the exhaustion and the particular matters, I just took a very old slide from our own studies where we looked on how the impact of airborne pollutants with the example of smoking, how that can affect the lungs, but in individuals with particular genes, it may start immunity. In our case, again, citrinated antigens, but in many other diseases we have now known that this type of things happening in the airways can also trigger immunity that may lead to a myositis, that may lead to lupus, that definitely will lead to multiple sclerosis. So the lung is a site which very often is a site where immunity leading to autoimmune diseases is being triggered. So that was the reason why we set up a study and analyzed what happened in this case for RA. I cannot cover all diseases. From air pollutants, including those which are produced by fossils and leading to climate change. So how we did it is that we have, in our country, we have population-based registries, in this case for rheumatoid arthritis. We have made a very big case control studies going out from newly diagnosed cases of rheumatoid arthritis. We've asked a lot about professions, about occupations, but also amongst lots of other lifestyle factors. And we have asked exactly the same questions to very carefully selected population controls. We now have about more than 5,000 individuals as cases and similar numbers of controls. So this very complicated slide shows how we did it, because we were interested in how was the impact of agents affecting the airways in different type of professions. And then we asked which type of profession, which type of occupation had all these individuals, 5,000 in different cases, 5,000 controls, had over their lifetime. This is what we had to the left, occupational information in ERA. Then we made what is called a job exposure matrix. That means that you can say which are the exposures, in this case for inhalable agents, if you have had particular type of occupations and professions in our country. And then you put them together, so you could then see what is the likelihood of having been exposed to different agents if you have had these occupations. And then we could make an index for each type of exposure for each individual in this case control study. And that enabled, in this case, Bao and Tang and Zhang, very gifted students and postdocs in our lab, in our unit, to investigate what happens with many occupational agents. And this is also a complicated slide. You see that to the right in the heat map, you see that many of them go together. But if we isolate them and look to the left, we could just say that go from the air, we have detergents, we have carbon monoxide, we have stone and concrete, iron, we have lots of combustion, diesel engines, and lots of other things. And what we see is that more or less all of them have an impact on RA overall. The impact is larger in agreement with what we have found for smoking for the aqua-positive disease, but there is also an impact for the aqua-negative disease. If we continue and look for the dose-response relationship, we see that if you are exposed to, in this case, up to 5 different exposures, then the risk for having, in this case, the RA is about 2.2, It goes out to 2.7 if you have aqua-positive disease, but you see it also there for aqua-negative disease. Then we could add genetics. In this case, they used what is called a genetic risk score. That is putting together all the known genetic polymorphisms, which are indicated here by red arrows, and making a score of their relative importance. And then you could divide the population in those half who have a relatively high score and those half have a lesser score. Doing that and combining these things, and this is now one of the critical slides showing that if you look, for example, to the right, with quartz dust, but it goes the same for gasoline engine and for the others, you see the first bar is just the reference. If you are not exposed to these agents, you are not a smoker, and you do not have the high risk score. The next is if you have the occupation. The next is if you are a smoker. The next is if you have the genetic risk score. But the interesting thing is if you have all these three, which half of the population thus has, to the right, the black bar for each of these exposures, you see that the risk figures go up to 30. It's not the type of things which we are usually seeing for genetics with 1.2, 1.4, 1.5. It's 30 if you have these three things being exposed. And all these things are, of course, the environment related to different type of environmental agents, which are particularly emissioned during climate change. We could continue and looking for what happened with, in this case, diesel engines, which is very much the same as other type of gasolines. And you see here again, with the pluses to the left, that if you have all three exposures, in this case you have 16 times risk of getting aqua-positive disease, and a moderately increased risk of having aqua-negative disease, only 2. So the summary here is that, and then we also have the same thing for building workers, in this case with stone and concrete workers. And you see also here, you could see then you summarize the 3 risk factors, you have an increased risk odds ratio of 15. So the summary of this is that the results of fossil burning and other things, of course, because also other types of particulate matters, which of course in this country come from household burning and from other sources, have a major impact not only on the respiratory tract diseases, which is obvious and was obvious from the Lancet report, but also in this case for rheumatoid arthritis. And what we know is that will be very similar to several other rheumatic diseases which have not been investigated as closely as rheumatoid arthritis for these type of exposures. This shows the opportunity. I also downloaded the number of publications in the world related to climate change and health. And you see that there is a rapid awareness of these problems in the scientific community. However, when I made the same search in PubMed for climate change and rheumatic diseases, I only got 14 hits. So this means that there is a large opportunity for learning more of what happens, and in particular time to go what I talked about yesterday, namely prevention of these diseases, which I think is the most efficient of all things that we can do for our patients and those who should not be our patients. And I'm happy to hear from Dr. Kumar from Delhi that you actually in India you have a program for climate health, for climate change and health from your departments, and she is an active member of that program, I heard. So we would be very happy to work together in these very urgent things which affects all of us in the world. So my conclusions are that The driving force behind this climate change, the excessive use of fossil energy worldwide, has a lot of impact, of course, on human health. For example, this is just an example on the exposure to lines of noxious substances, both directly, as I tried to indicate for RA, and indirectly, of course, from the global change, global warming, infectious diseases, etc. And of course, as scientists and as part of the global community of sciences, we have to consider what is the impact of this, not least, as said from the beginning, from the young people who are sort of concerned with all rights about their future. So we should, of course, as scientists, try to follow these young people in awareness of these diseases and what are the impacts. And I'm happy to have help with, not least from the Department of International Health in Stockholm, where one of my friends, Hans Rosling, was a prominent contributor to the discussion worldwide. So, thank you so much.
Thank you, sir, for the excellent presentation. I would like to welcome Dr. Vit D to kindly present his talk.
Right, thank you. I understand this is not the right time to talk. Postprandial always has a bad effect on the memory. Go back. The topic that has been given to me is vitamin D, and we call it the master orchestra player in the rheumatic musculoskeletal diseases. I love this molecule because I've been dabbling with it for the last 5 years. It is very cheap, affordable, and therefore it can actually help a lot of poor people suffering from this disease. The bottom line is we are very much ignorant about this molecule, and there are lots of myths and facts moving around. And if it is really successful and we know how to find out its potential, true scientific potential, it's bad news for big pharma because of the cost it's entertained. Now, who is called a market orchestra? The conductor who actually understands the pulse of every player in the team to produce what is called the master symphony, the perfect music. And vitamin D has that ability and understand the physiological processes of different systems. And this is why we say that it is a master orchestra, but it is not known well. Nobody perhaps has heard the full detail of the orchestra that it is playing. Now, why it is so important? Let's understand evolutionary phenomena of this molecule. It is a very, very ancient molecule, and it has a plethora of biological activities. Do we know them? It has excellent cellular metabolism. It controls cellular metabolism. It affects the growth and differentiation of the cells. It stops apoptosis, helps in reproduction. Multiple papers are there. We know only this, and we have been taught all along that vitamin D is necessary for calcium homeostasis and skeletal health. True, that's what we are taught in the medical school. Until now, everybody talks about skeletal health, preventing fractures, preventing this, elderly's need. Causes remyelination of the brain, causes intestinal barrier function in colon, so that it has a wonderful role in preventing IBD. And most important for us, it's an excellent immune modulator. Excellent immune modulator. But unfortunately, we are stuck with the calcium homeostasis and its metabolism. That's where we make a mistake. And that's why people don't know much about this molecule. Now, whenever I go and speak about the importance of vitamin D in RMDs, there's a big laugh. And they say, oh, it's just a vitamin, not a biologic or a JAK inhibitor, dude. What are you talking about? Is a vitamin is a vitamin. How can it do so many things? As I showed you earlier, the extensive action it has on the different systems of the body. People have been working on vitamin D in aging, cardiovascular disease, brain diseases, lungs, infections, name it. But unfortunately, it is still labeled as a vitamin. It's a secosterol. It has almost steroid-like properties, but provided it's given in the right dosage. The reason for this ignorance is mass-scale deficiency or insufficiency in the world. It's a global pandemic. We understand now because I think we are measuring vitamin D widely. I think every laboratory, every rheumatologist is asking for a vitamin D without knowing why he has ordered that. And nearly 1 billion people are affected globally. India is no exception. And there are pooled 97 studies from India which has come out. Almost 56 to 65% of people have low vitamin D. It must be more. Whether it is under 18, more than 18, you can see the range is 67, 70, 80, depending upon which kind of population you are dealing. But the overall message is we are all insufficient or deficient in vitamin D. And what does it mean? People think, okay, if you have vitamin D low, take it, it will prevent fractures. Perhaps it will prevent fall in elderly. There are several variables affecting synthesis and levels of vitamin D. That we must know. When we analyze the levels, we must know that instead of just giving vitamin D to every patient who has low levels. Age has an impact. The extremes of age has low vitamin D. BMI, obese people, there's so much of obesity in the world. Obese people have low vitamin D because dissolved in the fat cells. Geographical location, people 35 degrees above latitude have low vitamin Ds because of low exposure to sunlight. That is what I was talking about. Sunshine, vitamin. If you get exposed properly, nowadays nobody wants to get exposed to sunlight because they don't want to get dark, women mostly, and therefore the exposure is very less. Winter season, you get low vitamin D. If you have dark pigmentation, you need to be exposed for a longer period of time to get adequate dosage. There are drugs like steroids, like anticonvulsants, which can cause low vitamin D. And of course, genetic mutation affecting the receptors, the vitamin D receptors. If there is a mutation, then it becomes low. So whenever we see a prescription or we see a report from the lab, these are the things we must keep in mind, that whether we are analyzing it properly before we say, okay, he's vitamin deficient, insufficient, and for this reason, and that is what we need to treat. That I say is ignorance. That is where we fall to understand. We fail to understand. All of us know about the basic metabolism. 90% of our vitamin D comes from the sunlight and about 10% from food. And there are a lot of vegetarians who obviously not get adequate vitamin D. Food is mostly fish, eggs, things like that. But 90% is from the sunlight. UV light falls on the skin. On the 7-dehydrocholesterol, it gets converted to 25-hydroxycholesterol, 25-hydroxyvitamin D in the liver. And from the liver, it goes to the kidney where there is 1,25-dihydroxylation, and that is the active form of vitamin D. But mostly we measure 25-hydroxy. And therefore, it has been found the status of vitamin D is based on 25-hydroxy. So there is a problem here. Very difficult to measure 125 because it is not very stable to analyze. And this goes and sticks to the vitamin D receptors wherever they are present. It is almost ubiquitous, VDRs, because evolution has a purpose for this vitamin. And then it goes to the different systems. It has endocrine, paracrine functions, antibacterial, antiviral functions, anti-inflammatory functions. But we know the endocrine functions of increase of stabilizing calcium metabolism and increasing skeletal health. How does it work? As I told you, it's a secosterol. It goes into the cell, it gets attached to the vitamin D receptors, and then it causes heterodimerization with retinoic acid receptors. Goes and sticks to a promoter molecule called vitamin D receptor responsive element, which is the promoter for lots of genes. Now imagine this vitamin regulates so many genes in your cells. You can understand why it's called a master regulator. and then the whole gene expression occurs, including epigenetic changes. Why is vitamin D receptor ubiquitous? There has a purpose. We don't know the purpose. We know it's there, and nature does not allow dead wood to remain within our body. Nature always has a purpose for every molecule it gives you. These cells are extremely well distributed, but most important for us, for the rheumatic disorders, they are the innate immune cells and the adaptive immune cells. You have lots of vitamin D receptors activity in both the dendritic cells, monocytes, macrophages, T cells, B cells. They all contain a lot of vitamin D receptors. So therefore, if you do not know the status of vitamin D receptors, that it can undergo a mutation that can give rise to poor function of the vitamin D, irrespective of how much vitamin D you pump into the system, then obviously it's not going to give you good result. This is one of the studies that we did. We are looking at SLE, because I have a strong conviction that vitamin D is a very good adjunct in SLE, provided you know what is the dose that should be given. And we found a lot of association of vitamin D polymorphism. There are 4 polymorphic forms, 2 of them beautifully associated with increased incidence of SLE. So one has to know about this mutation as well when we analyze the system. Do we know the right levels of vitamin D? It's for skeletal muscles, that's why Endocrine Society defined vitamin D deficiency as less than 20 nanograms, insufficiency as 21 to 29, and normal for 30. And this 20 has come where the parathyroid hormone is no longer stimulated. We call it normal. But is it the real amount that we need for rheumatic musculoskeletal disease? No, we don't know that. That is why we have a problem in analyzing and saying whether vitamin D is useful or not. Now, the first thing is the genetic, the basic, the molecule that goes and controls the genes of a body must be a very important player. It affects almost 1,000 genes in your body, thousands. Some of them go up, some of them go down. I cannot narrate the large numbers, especially the immune modulatory genes. With anti-inflammatory genes comes down, the pro-inflammatory genes go up, and now you give vitamin D, the pro-inflammatory genes will come down, the anti-inflammatory genes will go up. As you can see this is called the heat map, expressing 1 column expresses 1 gene. And you see, as you increase the amount of vitamin D, the changes start occurring. If you look at individual cells, whether it's epithelial cells, fibroblasts, B cells, granulocytes, monocytes, muscle cells, T cells, many genes are upregulated or downregulated based on the kind of environmental factor, based on the kind of diseases that we face, and therefore it is so important that this is an important factor in our body. The second most important thing is immune modulation. The dendritic cells are the bridge between the innate and adaptive immune system. You control the dendritic cells, the polarization of the naive T cells get affected. If you have low levels, you get high levels of Th1, Th17, which we don't want because we know that is linked with autoimmune diseases. But if you have good amount of vitamin D, the dendritic cells get modulated, and you have downregulation of the Th1, Th2, and upregulation of Treg cells. But the bottom line is how much? How much? That we don't know. Individually, if you look at the innate immune system, you can see the macrophages are affected, and that is where it helps in preventing infection. It causes increased amount of cathelicidine, defensin, IL-2, and downregulates TLR2 and TLR4. And the dendritic cells, it prevents co-stimulation molecules to be expressed and antigen presentation to occur, and inhibits inflammatory cytokines. Vitamin D was a very, very important molecule during COVID infection. It was found those who have high vitamin D, above 50 nanograms, were protected against severe COVID infection. Individually, individual cells have extremely important vitamin D function. The Th1, Th2 is downregulated, as you can see. The Treg cells get upregulated, and the B cells, there's inhibition of IgM and IgG. So you have 2 major systems in the body which is being regulated by vitamin D. One is the genes, other one is the immune system. I wrote an editorial that was way back in 2016, the unexplored immune modulator. I believe it's a wonderful immune modulator. Only thing we need to know more about it. Now, the concept, the proof of concept must come from experiments, not from physiological things that I showed you. Whether this master player of the orchestra is playing Bach or Beethoven, or is playing trash, first part is look at the mouse models As you can see, the rheumatoid arthritis mouse model, that the collagen-induced arthritis or systemic lupus, inflammatory bowel disease, autoimmune thyroiditis, vasculitis, vitamin D levels have been reduced, supplementation has been given, and there has been excellent improvement in all these conditions. Or you take a mouse, you do a gene knockout, you knock out the VDR, which is very important, as I told you, the receptor, or you knock out the 1-hydroxylase, that is CYP27B1, which causes the final active metabolite. The same thing happens. You get severe disease, and therefore the link is almost confirmed through animal experiments. But is that so in human subjects? The story goes like this. You look at all the systemic review and meta-analysis, whether it is RA, SLE, Sjogren's, all of them have shown very low or insufficient vitamin D. It's expected because even normal people have very low. And the outcome is in RA, is related to inverse correlation with disease severity. In SLE, it is inverse correlation with SLEDAI. In Sjogren's, associated with severity of dry eyes. And people have supplemented by 8,000, 50,000 per week, 12 weeks, 18 weeks, 6 weeks. I mean, there are different supplementation regimens, and they found improvement in DAS28, DSR, TJC, but moderate evidence. In SLE, they found reduced fatigue, but there was no affection with SLEDAI. And in Sjogren's, they found useful in improving dry eyes refractory to conventional treatment. This study we did in 2014, and we demonstrated in vitamin D levels in Indian systemic lupus erythematosus patients was closely associated with, inversely associated with disease activity and interferon alpha. We know interferon alpha is one of the most important molecules that drives systemic lupus. If you have a cheap molecule in a junk that downregulates interferon alpha, we don't have to give anifrolumab, which perhaps will cause some lax if it comes to India. That's why I told if vitamin D comes out to its potential, it is bad news for big pharma. More evidence, SPA, SSC, inflammatory myopathies, FMS, you can see invariably you'll find all these studies have shown low levels, which I'm not very surprised. Some of them found inverse association with some of the diseases. In SSC, it was high prevalence of osteoporosis, but no association with clinical phenotypes. In inflammatory myopathy, it was in deficiency associated with anti-Jo and anti-MI. Explanations are not given, but most of them do not have any randomized controlled trials to prove. That is where we have a problem. This is in 2022. All along we have been taught that vitamin D prevents fracture. This was supplemental vitamin D and incident fractures in middle life and older adults. This is what they did. This was a study done in US, and you found that supplements of vitamin D, that is 2,000 per day versus placebo, did not reduce the risk of bone fractures among midlife and older adults in the United States. The concept that we have all been carrying and giving vitamin D to elderly and other people, saying that it will prevent fractures, perhaps fracture is not dependent on simple BMD. Fracture is dependent on many other factors, the microarchitecture, the strength, which we cannot measure. BMD you can manage by DEXA, but you cannot measure this. Vitamin D increases your BMD fine, but it certainly does not help in preventing fracture. This is a randomized controlled trial published in NEJM in 2022. Again in 2022, this I think is a very, very good landmark trial. It's a vitamin D and marine omega-3 fatty acid supplements on incident autoimmune disease called vital randomized controlled trials. What did they do? It was done across the United States. 25,000 participants given 2,000 international units per day or masked placebo, and along with omega-3 fatty acids. They were followed up for 5 years. What was the result? Those who took vitamin D within 5 years, they found that the incident autoimmune diseases that occurred was lowered by 22%, irrespective of omega-3 fatty acid. If you don't even give omega-3 fatty acid, it does not make any difference. People who took only omega-3 fatty acid and not vitamin D, the reduction was moderate and was not statistically significant. So this is a very, very important evidence that vitamin D does play a very important role in RMDs. My summary is very precise. We know it's a master regulator because it controls a lot of genes, as I showed you, over 1,000 genes, maybe much more. And there's excellent data demonstrating its potency in modulating the innate as well as the adaptive immune system. Very precisely, people have found out very robust data. And we have got beneficial effects of vitamin D supplementation in animal models of most of the diseases that I showed you. And we have evidence that across the world there is a pandemic of vitamin D deficiency, low serum 25D, therefore, is associated with the increased prevalence and severity of autoimmunity. It could be just an association because everybody is deficient. But the crucial thing that I would like to emphasize, the missing piece of the jigsaw puzzle, is the absence of robust, randomized, controlled trials of vitamin D supplementation in humans. I just showed one vital trial which showed definite reduction in incident autoimmune disease by 22%. And therefore, what we need is that. And the second most important issue that needs to be debated is not clear what dose of vitamin D and serum levels of 25D are optimal in controlling RMDs. It's a huge possibility of looking at this molecule in a bigger way and trying to do more randomized controlled trial and trying to find out what is the optimal level of vitamin. People are very scared of vitamin D. It'll cause hypercalcemia, it'll cause this, it'll cause nephrolithiasis. It doesn't happen. It doesn't happen. I've tested over 10,000 patients, nothing happens. And only when it's more than 150 nanograms, there's toxicity. So we need to know what is the level, and I'm sure we'll get excellent result by administering vitamin D as an adjunct, provided we know what is the dose necessary to regulate some of the diseases. Thank you very much.
Thank you, sir. Now the session is open for the question and answer.
Well, we had two brilliant presentations on very important issues of climate change, and we know that apart from lifestyle and genetic predilection, now we are realizing that environmental changes are very, very important, and we have fossil fuel and O2, ozone layer depletion, carbon monoxide, carbon dioxide, SO3, what have you, all sorts of particles are there. So it is very important that we realize the importance of that. And I would like now open the house to question and answers.
Yeah, hello. I'm Dr. Roy from Hyderabad. My question is towards Vit D, sir. Sir, which formulation of vitamin D would you recommend to your patients apart from parenteral?
Sorry, I didn't get your question.
Which formulation of vitamin D would you recommend to your patients apart from parenteral therapy?
Yeah, there are capsules, there are sachets available, cholecalciferol, and there are nano particles. I've tested all three, and I found the nano preparation is the best.
Okay, so it has the best bioavailability.
We have been giving nano preparations, liquid nano preparations, and the absorption is much better than if you give it in oral capsule form or in sachet form.
Fine, thank you.
But I can't tell you which brand is better. That I can't tell you because I've not done that.
Any more questions? Yeah.
So I have a question on vitamin D, and that is that the study from Karin Kostenbader was done in the rather northerly and sun-poor, the climate of very little sun in the Boston area. They presumably have much less vitamin D than you, living in a much more sunny place in India. So my question is, have you compared the levels of vitamin D before the supplementation in Boston as compared to those which are here? Because that may affect the effect of the addition of vitamin D.
There are multiple confounders, as I showed you, and we have not done that study. We have not done that, so I'll not be able to tell you exactly what effect those variables or parameters have had on the levels of vitamin D. I don't have that data.
Any more questions? Yes? Then I think we'll move on to the next session. No, there is, yeah.
So, good afternoon everyone. I'm Dr. Rudrarpan Chatterjee from SGPGI Lucknow. and I'll be presenting on how real is the effect of UV rays and seasonal variation in RMDs. So I have no disclosures, and these are some key references to my slides. So India is a diverse country, not only its population, but also in its climate. And we have multiple different climatic regions in the country. So one such climatic region is the tropical region, where I started my journey in medicine in Mumbai. And a curiosity was that we didn't see much ACLE. We saw a lot of severe manifestations of lupus, like nephritis and NPSLE, but very little cutaneous lupus. 2019, I moved to Lucknow, and in the cold winters, suddenly there was this burst of cutaneous lupus activity that we saw. And what intrigued me was these almost textbook images of ACLE lesions in the phalanges. So I went back and asked these patients their daily routine. And they said that there's a very, very unique cultural phenomenon. In the cold winters, the patients, they go up on their roofs and they essentially sunbathe in the afternoons to feel warm. And at night, they sit around a wood fire and they warm their hands. So what's common there is the exposure to UVB that was not there in my previous place. So I'm sure I've induced some skepticism about that, because that's an anecdote. And what I propose to do in the next part of the talk is to substantiate this anecdote with some data. So UV rays are not homogeneous. There are three types, A, B, and C. UVC we don't need to worry about because it's filtered out by the ozone layer. UVA is interesting because it's actually immunomodulatory. It penetrates to the dermis, kills off B and T cells, and reduces inflammation and inflammatory cytokines. UVB is the most erythemogenic. It is 1,000 times more potent than UVA, inducing erythema, and it increases tissue injury and inflammation. So back in the 1970s, there was a flurry of activity in the published literature regarding this test called the Lupus band test. And this is essentially looking at UV rays and how they affect the skin. On a skin biopsy, when you do a direct immunofluorescence, you see this granular deposition of IgG and C3 on the dermoepidermal junction. And what was found was, if you did this Lupus band test in light-protected skin, and it was positive, then that predicted more severe lupus nephritis and worse prognosis. So 5 decades down the line, we now have some elegant data from mouse models that further substantiates this. So this is one such lupus-prone mice. And these mice have now been given a single dose of UVB radiation. And what we see is that the interferon response in the skin goes up manyfold over non-exposed mice. And this interferon response is more in females compared to males. And this is just a confocal microscopy image showing the same thing, the red being the interferon response. So does this apply to humans? And it was found that it does. So these 5 healthy human volunteers came up, and they were again exposed to UVB. And on doing a skin biopsy and a genetic analysis of the skin biopsy, you saw that again the interferon response was increased in the skin at 6 and 24 hours post-exposure. So does this translate to autoimmunity in the systemic circulation? And the answer again is yes, it does. So blood interferon scores from the mice, not from the humans, and kidney inflammation scores, again interferon signatures from the kidney tissue, show that there's a significant increase in interferon signature in the kidneys as in the blood. Now we have made a magic rabbit appear out of a hat. To prove its magic, you have to make it disappear. And that's established by HCQ. So when you pre-treat these animals with hydroxychloroquine before exposing them to UVB, they no longer develop the interferon response. So how is UVB being sensed by the cells of the body? It's actually the DNA damage that it's inducing. So it oxidizes DNA, and this oxidized DNA is picked up by something called a C gas, which is a molecule in the cytoplasm. And the C gas further stimulates STING, sting, and the C gas sting pathway downstream, it upregulates interferon beta production. So what happens when you knock out C gas in these mice? So knockout C gas mice have a subsequently reduced interferon response in the skin as well as in the blood. So is this relevant for clinical progression? And for this, there's a small cohort of at-risk individuals of autoimmunity. So patients, healthy people who actually have ANA positivity with at least one clinical criteria for lupus, but the symptom duration is less than 12 months and they're treatment naive. When they were exposed to UV radiation, again a single burst of UV light, they developed a 28.7-fold increase in the interferon response in the skin and a 2.2-fold increase in the response in the blood. And some of these patients actually went on to develop lupus in the future. And then they had a 479-fold increase in interferon response in the skin and 7.8-fold increase in the interferon response in the blood. So there's a definite gradient from healthy individuals to individuals at risk to individuals who actually develop SLE. And this is induced by UV radiation. So this is not the only mechanism in which autoimmunity is induced. It's well known that keratinocytes, when exposed to UV radiation, they form these apoptotic blebs on their surface. And on probing these apoptotic blebs with monoclonal antibodies, it was found that they contain antigens of Rho and La. And these are autoantibodies to Rho and La that are well known to cause photosensitivity. So if we move on to some clinical data, so this is a Canadian cohort of 258 patients. At the time that they developed incident SLE, it was evaluated the risk factors, what they had in the past year. And it was found that those who had a history of working outdoors for more than 20 hours a week or for more than 2 months a year in the past 12 months had a significant higher odds of developing systemic lupus erythematosus, 1.9 odds ratio. And those who were generally photosensitive, who tended to get sunburns, were again having a higher odds of 2.4 compared to those without. So outdoor work and tendency to sunburn is associated with patients of SLE. So how do we translate this to the clinic? Now, photoprotection is a well-known method to prevent autoimmunity or to prevent flares of autoimmunity. But I put a question here on the SPF 30. And the reason for that is your advice to your patients has to be culturally relevant. In our OPD, when a farmer who's working for 8 hours in the farm comes to me, if I tell them that you need to apply, every 4 hours you need to apply sunscreen while you're working, she'll not laugh at my face, but she'll be laughing inside, because that's not practical for her. What we can tell them is culturally relevant things, to use an umbrella, to wear full-sleeved clothing. This is something that they're doing 6 months a year, and for them to increase it to 12 months a year won't be a hassle. And lastly, I'd say empathize and reinforce, because we're trying to bring about behavioral change in our patients, and that is something that is very difficult to do in medicine. So if you're empathetic and listen to your patients will listen to you. So just to summarize, UVB spectrum of ultraviolet light does promote autoimmunity. It induces a local and systemic response, and that is an interferon response. UVB may promote autoantibody formation by exposing autoantigens, and it may be temporarily linked to progression of autoimmunity from healthy controls to at risk to SLE. And photoprotection, while essential, it should be practical. So I'd like to thank our patients and the institute that has given me the opportunity, and the IRACON organizers for giving me the opportunity to speak. Thank you.